Okay, good morning to one and all present. I welcome you all to the lecture on micronutrients. So, beginning with what micronutrient actually is, as the name suggests, micronutrients are called micronutrients because they are needed in very small amount. It does not mean that they are of less important than any other macronutrient. It is not like that. Micronutrient deficiency or toxicity can reduce plant yield similar to macronutrient deficiency or toxicity. They are simply called micronutrient only because of their amount is less required by the plant, not because of their less importance. And micronutrients called micronutrient because they are needed in less amount, and these substances are the magic vent. They are like koi jadoo ki chhadi hai that enable the body to produce enzymes, hormones, and other substances essential for proper growth and development. As we all know that there are 17 essential elements as per the essentiality criteria given by any one of you. Non-installed. Yeah. So there are 17 essential elements given by the essentiality criteria of Arnold Stout 1939 for the plant growth. And out of these 17 essential elements, 8 elements are considered as micronutrients. So it can simply be asked in exam how many micronutrients are there out of 17 essential elements. So you should have direct means you should not in that exam हम count करने नहीं बैठेंगे ना, it will take our time. So directly you remember that there are 8 elements that are micronutrients. And as on today, iron, manganese, zinc, copper, boron, molybdenum, nickel, and chlorine, they are included in this micronutrient category. As I said, that micronutrients are the nutrients that are required in less amount. But what is the exact that less amount? Quantitatively, we must know. So the concentration of these nutrients in the plant is found often within
फिनोमिना जो होता है, डिस्पर्जन वाला जो फिनोमिना होता है, जो हमारा रेनबो होता है, उसमें यही फिनोमिना होता है और सिर्फ डिस्पर्जन नहीं होता बल्कि उसमें इंटरनल रिफ्लेक्शन भी होता है. इस वजह से बोथ ए एंड बी सही होगा. ऑप्शन नंबर सी. ठीक है. अगला है एडवांस सनराइज एंड डिलेड सनराइज.
The concentration of these micronutrients in plant is found often within 100 mg per kg. What is this mg per kg? Any one of you? Is it ppm or ppb?
ppm.
Yeah. So the concentration of micronutrient in plant is often within 100 ppm, except iron and manganese, which can go normally up to around 500 ppm also. So generally we can say that micronutrients are those nutrients that are required by the plant in means it is less than 100 ppm. Okay, exception is there with iron and manganese that can go up to 500 ppm also. Now these elements, these are some of the other names of micronutrients that can be asked in objective questions. The micronutrients are also known as minor nutrients, trace elements, oligonutrients, oligoelements, or sperm elements. So you should remember these names at least. And out of 8 micronutrients, this iron, manganese, zinc, copper, and nickel, they behave as cations, and boron, molybdenum, and chlorine, they behave as anions in the soil. And the chemistry of micronutrient cations is different from that of micronutrient anions. So we'll discuss them one by one and we'll see how they are behaving differently. Now you see the elemental characteristics of micronutrients in the context of general trends in the periodic table. So if you see the atomic number of molybdenum 42, manganese 25, iron 26, nickel 28, copper 29, zinc 30, boron 5. You need not to remember all the information about these elements, but yes, at least you should know the atomic number plus their abundance in the earth crust. You must have read it in pedology section also that molybdenum in the earth crust is around 1 to 5 ppm. This manganese is 600 ppm, iron 3.5%, nickel 10 ppm, copper 25 ppm, zinc 71, and boron 15 ppm. At least you should remember this atomic number and their abundance in the earth crust. Another thing is, when we go from this molybdenum and to this boron, there is increase in the electronegativity. The electronegativity of this boron boron is highest if we see all these micronutrients, and this molybdenum is least. Now, before seeing the trend, let us first understand what this electronegativity means. If any one of you want to tell me what electronegativity means, you please proceed. If no, you simply say no so that I move ahead.
No, ma'am.
Okay. So electronegativity is the tendency of any atom of the element to attract shared electrons. So it is tendency of any element to attract electrons. कोई element electrons को कितना अपनी तरफ attract करता है, that is electronegativity. So this boron is having high electronegativity, means it is having the tendency to attract electrons. And this molybdenum is having least electronegativity if we see these elements. Now this electronegativity is affected by the atomic number. The element that is having least atomic number, you see this boron is having 5 atomic number. That means electrons around this boron are very less, no? So they are nearer to the nucleus. So less the atomic number and the distance at which these valence electrons reside from the charged nucleus. If the distance between the nucleus and the electron surrounding that atom is less, that means positive charge is relatively more, no? So electrons can be attracted to that particular element. So as the atomic number decreases, electronegativity increases. This is one thing. Another is acid character increases from molybdenum to boron. So boron is behaving more acidic and molybdenum is behaving basic in nature. So what is this acid character means? You see, acid character is the ability or tendency to lose a proton. So boron is having tendency to lose the proton easily, and molybdenum is having less tendency to lose the proton. So towards boron, this acidic character increases. You see, as we move from left to right in a periodic table, the atomic size of the element decreases. If the atomic size of the element decreases, which means electronegativity increase. जितना कम atomic size होगा, उतना ज़्यादा electronegativity होगी. Electrons को बहुत आसानी से वो attract कर लेगा. और अगर electrons को accept आसानी से कर रहा है, that means it is getting more polar. So it is easy, relatively easy to lose proton. So boron acidic character is more, and this molybdenum is behaving like a basic. So from this slide, what is important to remember is the atomic number of all the elements, their relative abundance in the earth crust, electronegativity behavior of all the elements. At least you should know the sequence, or agar wo bhi yaad nahi rakhna, so you should remember which one is more electronegative and which one is least. Okay, this is the least expected from you. Another is acid character behavior and basic character behavior. So this slide is for this purpose only, to get you the general idea of all these micronutrient elements. Now moving ahead. Geochemistry of micronutrients. If any one of you want to tell whether my speed is okay to all of you, or I should go a little slow.
Yes, ma'am.
It's okay, ma'am.
Okay, fine. So geochemistry of micronutrients, you see, trace elements, this is the other name of micronutrient only. Trace elements, content of the soil depends on the parent rock type. Which element is there in the soil? It depends on the parent rock, na? Because ultimate source of these nutrients in the soil is the parent rock and the weathering processes. We know that there are 3 kinds of rock: igneous, sedimentary, and metamorphic. And under igneous rocks, we also know that there is acidic rock, basic rock, ultra basic. So this basic igneous rocks have higher content of manganese, cobalt, nickel, copper, and zinc, whereas sedimentary rocks are rich in boron and molybdenum. And during the crystallization of the minerals in the rock formation, trace elements enter in the framework of the minerals. When there is crystallization of minerals and when there is formation of the rocks, these trace elements enter into the framework of the elements. And the position of these trace elements in the mineral framework, it depends on its radius. We know that mineral, there are different structures of minerals, no? 1:1, 2:1, 2:1, you must have read it in pedology section. So depending upon the size of the particular element, these trace elements can take position in the mineral framework, whereas its abundance in the mineral is decided by its chemical affinity. The element which is having more affinity towards, means if it is more reactive, then it will be more abundant in a particular mineral. So the position of the trace element depends on its radius, and its abundance depends on the chemical affinity. More affinity it is having, more will be the particular element in the mineral. And based on the affinity of the elements for the different minerals, trace elements are divided into 3 groups based on their affinity, right? So it is deciding the abundance of the minerals, right? So based on the affinity, we have 3 groups. One is lithophile, another is chalcophile, next is siderophile. Lithophile is one that is having high affinity for the silicate minerals. And the elements that comes under this category are boron, zinc, manganese, nickel, and iron. So they are lithophiles and they have high affinity for the silicate minerals. Another is chalcophile. They are the elements that are having high affinity for sulfide minerals. They form covalent bond with the sulfide mineral. Sometimes it is asked in exams that which one of the following group of elements form covalent bond with the sulfide mineral. So it is chalcophile. Examples are zinc, iron, molybdenum, copper, and nickel. Another category is siderophile. These are the elements that are having high affinity for iron minerals. Examples, iron, copper, molybdenum, and nickel. Another category is also there. Which one is left? Any one of you? Lithophile, chalcophile, siderophile. One more is there. Yes? Okay, fine. Another is, repeat please.
Hello.
I'm not getting you. You please speak it again.
Atmophiles, ma'am.
Okay, fine. So atmosphere is also one category, means the elements that are present in the atmosphere. But here we are talking about the affinity of elements to different minerals, right? So that is why atmosphere category I have not written, because we are talking about affinity to different minerals only. Okay. Now, moving ahead. Coming to different sources of these micronutrients, there are different minerals in the earth crust and they are the sources of micronutrient, and this slide is very important from exam point of view. In most of the times I have seen objective questions from this particular slide, that a particular mineral is given and you are asked which, means this particular mineral is a source of which micronutrient. Okay, so iron, we have hornblende, hematite, goethite, pyrite, magnetite, olivine, and this pyrite is other name of pyrite. Any one of you?
Fool's gold, ma'am.
Yeah, wonderful. And its chemical formula?
Marcasite, ma'am.
FeS2.
FeS2. Yeah, FeS2. Marcasite is also one of the
White
polymorph of pyrite. Yeah, but this pyrite is FeS2 and it is fool's gold. Okay, so this question can also be asked. So iron minerals are hornblende, hematite, goethite, pyrite, magnetite, olivine. Manganese minerals, pyrolusite, magnetite is also there, rhodonite. Any other manganese mineral if you know? Yes. Other than this pyrolusite, rhodonite, magnetite. Have you heard of rhodochrosite? Yes.
No, ma'am.
Okay, you write. Another manganese mineral you write, it is sometimes asked in exam. You write rhodo, RHODO, and CHROSITE, rhodochrosite. Another is hosmenite, HAUSMANNIT. So pyrolusite, magnetite, rhodonite, rhodochrosite, hosmenite, these are manganese minerals. Generally this pyrolusite is asked in exam, but yes, हमें थोड़ा-थोड़ा तो सबका idea होना चाहिए. Next is copper. So copper minerals are chalcopyrite, covellite, cuprite, malachite. One more mineral that is, I have seen in recent objective books that they are asking that particular mineral also. So if you want to tell me any other mineral of copper, any one of you, hint for you, it starts from the A, capital A.
Azurite, ma'am.
Yeah, wonderful. So copper minerals, you write chalcopyrite, covellite, cuprite, malachite, and azurite. Okay. Next is zinc. Zinc containing minerals are sphalerite, Smithsonite, hemimorphite is also there. So sphalerite, smithsonite, hemimorphite. Boron containing minerals, tourmaline, very common question. Colemanite, borax, kernite. Another is chlorine containing minerals. So it is apatite, muriate of potash, KCl, sodium chloride, NaCl. Molybdenum containing minerals, molybdenite, wulfenite, ferrimolybdenite. Another molybdenum containing mineral that is asked in objective question, you write it as powellite, POWELLITE, powellite. This is calcium molybdate, CaMO4. So molybdite, wulfenite, ferrimolybdenite, another is powellite. Nickel containing mineral, serpentine, olivine. This is very important table. So इसको ध्यान से देखना है, ठीक है? Now moving ahead. As we have seen in the starting slides, that there are certain micronutrients that are taken up by the plants in cationic form, and there are certain micronutrients that are taken up by the plant in anionic form. More specifically, there are 5 cationic micronutrients and 3 anionic micronutrients. So their ionic form, iron is taken up by the plants in Fe2 plus and Fe3 plus, ferrous and ferric form. Which one is ferrous and which one is ferric?
Fe3 plus is ferric.
Fe3 plus is ferric, yeah. So it is ferric and this one is Fe2 plus is ferrous form. Manganese in Mn2 plus and Mn4 plus. Copper in Cu2 plus form. Zinc, Zn2 plus. Boron is mainly taken up by the plants in boric acid form, in neutral, H3BO3. But yes, other forms of boron, that means that can be converted. That is H2BO3 minus. We'll see this one by one. So these are the different ionic form of boron. Now, chlorine is taken up by the plant in chloride. This is an ionic form. Molybdenum in molybdate ion, nickel, Ni2 plus. So these are the ionic forms of these micronutrients. Now, taking these micronutrients one by one, first one is iron. It is taken up by the plant as ferrous form, Fe2 plus form by the plant. In soil, ferrous and ferric form, Fe2 plus and Fe3 plus, both are there. But plant take up this ferrous form, Fe2 plus. And the concentration of iron in the range of 100 to 500 ppm in mature leaf tissue is regarded as sufficient for optimum crop production. So the sufficient concentration of iron for optimum crop production is 100 to 500 ppm. And since this iron is transition metal, so it exhibits 2 oxidation states, Fe2 plus and Fe3 plus. It is constituent of 2 groups of protein. One is hemiprotein, another is iron sulfur protein. So hemiprotein means it is one that contains iron in porphyrin complex. So hemiproteins are those proteins that contain iron in porphyrin complex. So what does this porphyrin means? Porphyrin means it is an aromatic macrocyclic compound. So it is a ring type compound that consists of 4 pyrrole units, 4 bridging carbon units. You must have read the structure of chlorophyll, I guess. There are 4 pyrrole rings and magnesium is in center, right? So porphyrin means this cyclic compound. So hemiproteins are those proteins that contains iron in the porphyrin complex. And the examples of these hemiproteins are cytochrome oxidase. These are the enzymes. Catalase, peroxidase, leg hemoglobin. Another is iron sulfur protein, and the example of this kind of protein is ferredoxin. So iron is a component of hemiproteins and this iron sulfur protein, that is ferredoxin. This iron activates a number of enzymes, and because of its variable valency, it is assigned the role of biological redox system. So since it is having variable valency 2 and 3 plus, so it is having role in the redox system. It plays a role in nucleic acid metabolism, and it is a structural component of porphyrin molecule, which is involved in oxidation reduction reactions in respiration and photosynthesis, and it is necessary for synthesis and maintenance of chlorophyll in the plants. Now, the plants that are having less than 50 ppm of iron are classified as iron deficient. Somewhere it was written that Fe2 plus critical level of iron deficiency means if the Fe2 plus concentration in the plant is less than 30 ppm, then there is deficiency of iron, right? So these are 2 statements. One is saying that if plant is having having less than 50 ppm of iron, it is iron deficient. Another is saying if it is less than 30 ppm of iron, then it is deficient. So if you are able to find out the difference between these two statements, you tell. Any one of you?
Ma'am, in first one,
Yeah.
iron may be in form of ferric.
Okay. Any other one to tell any difference if you are able to find out? Any one of you want to try? This is one thing that could happen, means if it is Fe3 plus and this is Fe2 plus. और कुछ कोई सोचना चाहेगा कि क्या हो सकता है पॉसिबल डिफरेंस दोनों में? गलत ही तो होगा, सोच तो सकते हैं ना, बोलो. Okay, fine. नहीं बोलना, ठीक है. So this is saying that if Fe2 plus, this ferrous, if it is less than 30 ppm, then it is deficient in plant. This one is not for ferric actually, this is for total iron. Ferrous and ferric both. Okay, this is for total iron. So if total iron content is less than 50 ppm, then it is iron deficient. And if ferrous specifically it is asking, it is less than 30 ppm, then it is iron deficient. Fine. Now the deficiency of iron results in interveinal chlorosis appearing first on the younger leaves with leaf margins and veins remaining green. So if iron deficiency is first appearing on the younger leaves, you tell whether iron is mobile or immobile in plant.
Immobile.
Yeah. So if deficiency is seen in the younger leaves, it is immobile. And another is Lyme-induced chlorosis. Because of the Lyme application, there is deficiency of iron and that induced chlorosis. So it is very common question in exam. Which of the following element causes Lyme-induced chlorosis? So it is iron. That means because of the application of lime, what does this lime do? What is the effect of lime on soil pH? Whether it increases or decreases soil pH?
Decreases soil pH.
Decreases soil pH.
Increases soil pH.
Okay, those who are saying that it decreases soil pH, you tell me whether lime is applied in acidic soil or alkaline soil. Yes?
Acidic soil.
Lime is applied in acidic soil. No? So if lime is applied in acidic soil, that means definitely it is increasing soil pH. That is why it is applied in acidic soil. No. So because of the application of lime, the pH of the soil increases. And if pH is increased, that means there is deficiency of this iron. I'll tell you what is the effect of pH on all the micronutrients, means cations and anions. This we'll see in the subsequent slides. But yes, for now you keep this information with you, that because of the application of lime, the pH increases. And if the pH increase, then there is deficiency of iron. And because of deficiency of iron, there is chlorosis. So that is why this lime-induced chlorosis is because of iron deficiency. Okay. Now moving ahead. And one more thing you can write over here in this lime-induced chlorosis, you write that soil iron concentration is high. We have seen when I was telling you about the concentration of these micronutrients, I told you that all the micronutrients are within the range of 100 ppm, but this iron and manganese, there is exception that it may go up to 500 ppm also, right? So soil iron concentration is high, but it can become unavailable for absorption if soil pH is higher than 6.5. If the pH of soil is higher than 6.5, even if the concentration of iron in soil is high, still it will not be available to the plant. So you remember the pH, more than 6.5, this iron, this lime-induced chlorosis will be there. Okay. Now moving ahead to another micronutrient cation, that is manganese. So it is absorbed by the plant as manganese form, Mn2 plus, and healthy manganese sufficient mature plant contains 20 to 300 ppm of manganese. It is a transition metal and it is present in plant in Mn2 plus form, but it is easily oxidized to Mn3 plus and Mn4 also. Okay. And it plays an important role in the photosynthesis and detoxification of the superoxide free radical. This is very important function of this manganese, that it plays role in the detoxification of superoxide free radicals. Now question comes, what is this superoxide free radical, right? So you see, it is primary oxygen free radical which is produced in the mitochondria via the slippage of an electron from electron transport chain to molecular oxygen. If we have a molecular oxygen, O2, right? And if this oxygen gets an electron, then there will be O2 and there will be electron, negative charge will be there with the oxygen, right? So this is free radical. So when primary oxygen takes up the electron from the electron transport chain to become a molecular oxygen with a negative charge, this is what we call a free radical, and it is unstable and it is highly reactive. So it is highly reactive. So what does it do? It reacts with the cell wall and degrade the cell. So it is very important to capture this free radical, and this work is done by this manganese. Okay, so manganese play an important role in detoxification of superoxide free radicals. This you remember. It can substitute for magnesium, and it is an integral component of water splitting enzyme which is associated with photosystem 2. This is also very important question, very frequently asked in exam, that which of the following micronutrient or which of the following element is a component of water splitting enzyme which is associated with photosystem 2. So it is manganese. As we have seen that it is constituent of superoxide dismutase. So it is manganese SOD superoxide dismutase, which being present in, it is also asked in exam that manganese SOD, manganese superoxide dismutase, is present in which cell organelle? So it is present in mitochondria, peroxisomes and glyoxysomes. Whatever way you want to remember, you remember this one. Manganese stored in mitochondria, peroxisomes and glycosomes. And it protects the cell against the deleterious effect of free radicals. It is a constituent of nitrite reductase and hydroxylamine reductase, and hence it is linked with nitrogen assimilation. So manganese is a constituent of nitrite reductase, hydroxylamine, superoxide dismutase, water splitting enzyme. These are the important functions of manganese. Now moving ahead. Manganese deficient plants contain less than 25 ppm of manganese. So if it is less than 25 ppm, it will be deficient in the plants. And the deficiency symptoms of manganese are more severe on the middle leaves than on the younger leaves, because manganese is preferentially translocated to the younger leaves. This is a mobile element in the plant. So it moves to the younger leaves, and hence its deficiency is not seen on the younger leaves like iron. So its deficiency is seen on middle leaves. These are the common deficiency symptoms of manganese. This you may find very frequently in exams. Gray speck of oat, specked yellow of sugar beet, marsh spot of peas, pahala blight of sugarcane, and frenching of tongue trees. So these are the deficiency symptoms of manganese. Okay, if any one of you want to tell me, what do we understand by this frenching? Anyone? Okay, fine. अभी next slide में बताती हूँ मैं Frenching means क्या है, but for now you keep this information with you that Frenching of tongue trees. Next we'll see Frenching of citrus. That is because of different micronutrient. So Frenching of tongue, Frenching is actually mottling. Okay, there is variegated. Some part of the leaf there is another color. Some part of the leaf is having another color. That is mottling. So Frenching of tongue trees is because of manganese. Okay. Now moving ahead to another micronutrient, that is zinc. So plant take up zinc as Zn2 plus ion, and the zinc sufficiency content in plant is 25 to 150 ppm, and the zinc does not have variable valence. It is only Zn2 plus. Hence, no role in influencing oxidation reduction processes directly. It does not influence oxidation means redox processes directly because it does not have any variable valence. It is constituent of three enzymes: carbonic anhydrase, alcoholic dehydrogenase, superoxide dismutase. You see, superoxide dismutase we have seen in manganese also. So that was manganese SOD. This is zinc SOD. Okay. So carbonic anhydrase, alcoholic dehydrogenase, superoxide dismutase. The constituent of these enzymes is zinc. And zinc is involved in the synthesis of indole acetic acid, metabolism of gibberellic acid, and synthesis of RNA. Very important function of zinc, that it is involved in the synthesis of indole acetic acid. This is oxen, no? So if there is deficiency of zinc, That means there is less oxygen. And if oxygen is less, the leaves or the cell will not grow. So because of the deficiency of zinc, the leaves are little, very little, small leaves are there, and it form a bushy like structure. This is what we call rosette structure. This is because of the problem in the synthesis of indole acetic acid because of the deficiency of zinc. So zinc is involved in the synthesis of indole acetic acid, metabolism of gibberellic acid, and synthesis of RNA. Zinc promotes synthesis of cytochrome also, and it plays an important role in the stabilization and structural orientation of membrane proteins. So it stabilizes membrane proteins also, and it zinc influence translocation and transport of phosphorus in the plant. You tell whether zinc and phosphorus are synergistic in nature or antagonistic.
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 Yeah. So zinc and phosphorus, very frequently asked in exam, they are antagonistic, and under zinc deficiency, excessive translocation of phosphorus results in the phosphorus toxicity. If there is deficiency of zinc, definitely phosphorus will be more, and there can be phosphorus toxicity also. Now, zinc deficiency, you see, if the plant contain less than 15 ppm of zinc, then it will be deficient. And common symptoms of zinc deficiency includes interveinal chlorosis, reduction in the size of the younger leaves, as I told you, which are often clustered and gave bushy rosette appearance. This is because of the less indole acetic acid production because of zinc deficiency. Different means zinc deficiency in different plant is known by different names. These are Khera disease of rice. This is very frequently asked in exam. I think in most of the exams they used to ask this Khera disease of rice is because of zinc deficiency. It was discovered by whom? You tell.
Vial
Nene.
Where? Pantnagar. So yes, in different ways this Khera disease is usually asked. This is due to zinc deficiency discovered by Vial Nene in Pantnagar. Another is white bird of maize, little leaf of cotton. Yeah, Frenching I told you. So Frenching means mottle leaf only. So Frenching of tongue trees was because of the manganese, right? And Frenching of citrus is due to zinc deficiency. That is why I emphasized more on Frenching in last slide. So that you remember. Frenching of citrus because of zinc deficiency, Frenching of tongue trees because of manganese deficiency. Another is rosette disease of apple. Now moving ahead to another micronutrient cation, that is copper. So copper is absorbed by plant roots as cupric acid, cupric ion, that is Cu2+. And copper sufficient plants, if they contain copper between 5 to 30 ppm, then they are sufficient. And if it is between 20 to 100 ppm, then it is toxic to the plant. Copper exists as Cu plus and Cu2+. So this is cuprous form, this is cupric form. Plant take up this Cu2+, cupric form. And this copper is a component of number of enzymes like diamine oxidase, cytochrome oxidase, superoxide dismutase. Zinc SOD we have seen. So this copper and zinc SOD, they are together actually. So copper, zinc, superoxide dismutase. This is located in cytoplasm, mitochondria, and chloroplast. I'm emphasizing this again because they use in objective question they used to ask what is means where is this manganese superoxide dismutase present? 3 organelles I told you. And this copper zinc salt is present in another 3 organs that are cytoplasm, mitochondria, and chloroplast. So this is important, you remember this. It is also a component of polyphenol oxidase. This copper is a component of polyphenol oxidase, which is involved in lignin biosynthesis. You're right. This polyphenol oxidase is involved in lignin biosynthesis. So it can be asked in exam directly that which of the following element plays role in lignin biosynthesis? It is copper. It is important in imparting disease resistance to the plant. This copper is a micronutrient that is important in imparting disease resistance to the plant. If I ask you which of the macronutrient impart disease resistance to the plant, you tell.
Potassium.
Potassium. Wonderful. Yeah. So in micronutrient it is copper that is imparting disease resistance, and in macronutrient it is potassium that is imparting disease resistance to the plant. This copper enhances the fertility of the male flowers also. Another is if the plant contains less than 5 ppm of copper, then it is deficient. And the deficiency of copper causes male flower sterility, delayed flowering, and senescence. Another symptoms or another disorders because of this copper deficiency are white tip, reclamation disease, and diabetes. So this is very frequent question in exam, that reclamation disease is caused by which of the following element deficiency. So it is copper. So what is this reclamation disease? Any one of you want to tell? You must have heard that copper deficiency causes reclamation disease. So what is this reclamation disease? ऐसे नाम पढ़ने से भी कुछ समझ आ रहा है क्या? Reclamation means? ट्राई कर
यस
सकते
मैम,
हैं
इससे किसी चीज को सुधार करना, चेंज दे
हाँ
रहा है।
ठीक है, मतलब कोई भी रिक्लेम करे, किसी भी चीज के लिए रिक्लेमेशन करेंगे तो कॉपर डेफिशिएंसी हो जाएगा क्या? या फिर कोई पर्टिकुलर स्पेसिफिक रिक्लेमेशन की बात कर रहा है? थोड़ा सोचो।
Reclamation of sodic soil.
Okay, one is saying sodic soil. Yes, another one you say?
मैम व्हेन एसिडिक सॉइल में अपन कैल्शियम का एप्लीकेशन करते हैं तो पीएच इंक्रीज करता है, तो इसकी डेफिशिएंसी हो जाती है कॉपर की।
पीएच बढ़ने से कॉपर की डेफिशिएंसी हो जाती है। ओके, पीएच बढ़ने से तो सारे माइक्रोन्यूट्रिएंट की डेफिशिएंसी हो जानी चाहिए, तो सभी माइक्रोन्यूट्रिएंट के लिए हम रिक्लेमेशन डिसीज कह दें क्या? ठीक सोच रहे हो सब लोग? ठीक सोच रहे हो? थोड़ा सोडिक वाले ने थोड़ा नहीं सोचा सही। एसिडिक सॉइल में लाइन डालने से पीएच बढ़ेगा तो उससे कॉपर डेफिशिएंट हो जाएगा, दैट इज वेरी करेक्ट। बट दिस रिक्लेमेशन डिजीज, इट इज अ नेम गिवन बिकॉज दिस कॉपर इज हैविंग वेरी हाई एफिनिटी फॉर ऑर्गेनिक मैटर। अगर हम किसी सॉइल में रिक्लेमेशन करने के लिए FYM डालते हैं, ऑल ऑफ यू अंडरस्टैंड हिंदी ना?
यस मैम।
हाँ,
यस
तो अगर किसी भी सॉइल में हम रिक्लेमेशन के लिए FYM डालते हैं, ऑर्गेनिक सोर्स डालते हैं, एंड दिस कॉपर इज हैविंग वेरी हाई एफिनिटी फॉर ऑर्गेनिक मैटर, सो अगर हमने FYM डाला तो सारा का सारा कॉपर उसमें बाइंड हो जाता है और उसकी वजह से कॉपर डेफिशिएंसी हो रही है। सो दिस स्पेसिफिकली इज सेड फॉर कॉपर बिकॉज कॉपर इज हैविंग हाई एफिनिटी फॉर organic matter, right? So reclamation disease, it is a disorder that affects crop growth on reclaimed land, which is caused by the deficiency of elements like copper. Making sense to all of you?
यस मैम।
Now moving
मैम
ahead.
प्लीज रिपीट।
Okay, you see reclamation disease मतलब हम किसी भी सॉइल को रिक्लेम कर रहे हैं, उसमें कोई प्रॉब्लम है उसको हम करेक्ट कर रहे हैं, ठीक है? और करेक्ट करने की वजह से कॉपर डेफिशिएंट हो जाए, तो अगर हम किसी भी सॉइल में FYM डालते हैं या कोई भी ऑर्गेनिक मैटर डालते हैं तो कॉपर बहुत इजीली, बहुत टाइटली इस ऑर्गेनिक मैटर से बाइंड हो जाता है, क्योंकि कॉपर की एफिनिटी ज्यादा है ऑर्गेनिक मैटर के लिए। इट इज हैविंग हाई एफिनिटी फॉर ऑर्गेनिक मैटर। सो हमने FYM डाला सॉइल में, कॉपर बाइंड हो गया FYM से और प्लांट को नहीं मिल पाया, जिसकी वजह से कॉपर की डेफिशिएंसी हो गई। मेकिंग सेंस?
यस मैम, थैंक यू।
Now moving ahead to another element, that is nickel. So this is the 17th element recognized as essential for the plant growth, means the latest essential element. Who has discovered the essentiality of nickel? Any one of you?
Brown 1987.
Do you know the other names of Brown? Brown के साथ दो लोग और भी हैं? Any one of you?
Brown, Wills, Carey.
Brown, Welch and
Carey.
Carey. Wonderful. And year
87.
1987.
Yeah, why I'm emphasizing this, because sometimes we all know when we have started this essentiality, who has given the essentiality of particular nutrient, तो हम कोई एक साइंटिस्ट ही पढ़ते हैं, लेकिन अगर ऑब्जेक्टिव क्वेश्चन ऐसा हो कि वो एक मतलब ऑप्शन में दो साइंटिस्ट हो, फिर ऑल का भी ऑप्शन हो, बोथ का भी ऑप्शन हो, तब क्या करेंगे? तो भले ही याद एक ही रखें, लेकिन एक बार सारे लोग का नाम देखना जरूरी है, ठीक है? So nickel essentiality, it is the latest micronutrient that is being recognized as essential element by Brown, Welch and Carey in 1987. And plant nickel requirement is the lowest of all the essential element, that is less than 0.5 ppm. Nickel is absorbed by the plant as Ni2 plus. This nickel exists in Ni2 plus form, but it can also assume Ni plus, Ni3 plus form also. Okay. And the concentration of nickel in means the sufficiency range of nickel in plant is 0.1 to 10 ppm. This nickel, very important question, that it is a functional constituent of urease enzyme. This urease enzyme is involved in which process?
Urea hydrolysis.
Urea hydrolysis.
hydrolysis.
Yeah. So this is a functional constituent of urease enzyme, and this nickel is actually a cofactor You remember that nickel is a cofactor that enable the urease to catalyze the conversion of urea into the ammonium ion. This is a functional constituent. It is not saying that nickel is a constituent of this urease enzyme. This nickel helps in functionality of this urease enzyme because it acts as a cofactor. Okay, so nickel acts as a cofactor and helps in conversion of urea into the ammonium ion. And in free living rhizobium, rhizobium is bacteria or fungus?
Bacteria, ma'am.
Yeah. So in free living rhizobium, adequate nickel supply ensures optimum hydrogenase activity. So if there is optimum nickel, so it ensures good hydrogenase activity in rhizobium. Characteristic deficiency symptom of nickel have not been defined adequately, but yes, if in a system where urea is used as a sole nitrogen fertilizer, okay, you see there is in soil, we are applying urea as a single source of nitrogen fertilizer for foliar spray. And nickel supply is poor in soil. हमने यूरिया डाला और नाइट्रोजन का सिर्फ एक ही सोर्स डाल रहे हैं और उस सॉइल में निकल नहीं है. Then what will happen? यूरिया मिल पाएगा क्या प्लांट को?
No, ma'am.
No. So in the system where urea is used as a sole nitrogen fertilizer for foliar spray and nickel supply is poor, then lower urease activity causes urea toxicity to the foliage. Urease काम नहीं करेगा अगर निकल नहीं है तो, और यूरिया कन्वर्ट नहीं हो पाएगा अमोनियम फॉर्म में. So plant is not going to take that urea, but there will be urea toxicity to the foliage because of the lower activity of urease, and this leads to severe necrosis of the root tips. So if nickel is deficient in the plant and we are applying urea as a source of nitrogen, then there will be toxicity of the urea, and this leads to severe necrosis of the root tips. Making sense to all of you? Yes?
Yes, ma'am.
Yes, ma'am.
Okay, now moving ahead to another micronutrient, that is boron, which is taken up by the plant in cationic form or anionic form. You tell.
Neutral form.
Neutral form. So boron is taken up by the plant mainly as boric acid, H3BO3, and can also be absorbed in some of its anionic form also. Okay, usually it is taken as boric acid, but yes, we can keep this boron in anionic form of micronutrient also because it is absorbed in some of its anionic form. One is dihydrogen borate, monohydrogen borate, and borate under high pH conditions. And boron sufficient plants contain 10 to 200 ppm of boron. And you see, boron is neither a constituent of enzyme nor it activates any of the enzymes. So boron is not involved in any enzymatic activity, unlike any other micronutrient. So boron is neither a constituent of any enzyme nor it activates any enzyme. It is responsible for cell wall formation and stabilization. And this boron is involved in the movement of calcium in the plants. So boron is involved in the movement of calcium in the plants, and hollow heart in groundnut can occur when the shortage of boron limits calcium movement. So if there is deficiency of boron, then this deficiency of boron may also leads to deficiency of calcium. And hollow heart in groundnut, this is because of shortage of boron that limits the calcium movement. Okay, and this boron is involved in pollen tube elongation and pollen germination. And and you see, I have told you that because of the boron deficiency, there is deficiency of calcium, right? So you see, if there is boron deficiency in the plant, then there is decrease in the production of indole acetic acid. This boron is also involved in production of indole acetic acid, IAA. Okay, so if there is deficiency of boron, there is decrease in the production of indole acetic acid. And this decrease in the indole acetic acid is responsible for the induction of calcium deficiency. So you remember that boron is involved in indole acetic acid production, and because of deficiency of boron, there is less indole acetic acid, and because of that, calcium deficiency is there. This boron imparts drought tolerance to the plant. So if I ask you any macronutrient that impart drought tolerance to the plant? Yes, macronutrient.
Ma'am, potassium.
Potassium.
So This boron also imparts drought tolerance to the plant, and it also facilitates transport of potassium in the guard cell as well as the stomata opening. So if we are saying that boron impart drought tolerance to the plant, that means regular boric acid spray will help in mitigating harmful effects of drought. So question can be asked this way also, that which of the following spray would help in mitigating harmful effects of drought? So it is boric acid spray, because this boron is having the property that impart drought tolerance to the plant. Okay. Now moving ahead to the concentration of boron in the plant that causes the deficiency, that is 5 to 30 ppm. If it is less than this range, then there will be deficiency of boron. And boron deficiency symptoms in different crops are heart rot of sugar beet, browning or hollow stem of cauliflower, top sickness of tobacco, internal cork of apple, cracked stem of celery. I have tried to incorporate those deficiency symptoms that are very frequently asked in exams. So इनको ध्यान से देखना. Now moving ahead to another micronutrient, molybdenum, which is actually anionic micronutrient. So molybdenum is the only heavy transition metal. So out of all the elements, this is heavy metal, right? Molybdenum. So it is the only heavy transition metal which is taken up by the plant as molybdate ion. And the sufficient range is 0.1 to 2 ppm. If it is this much amount is present in the plant, then it is sufficient. And it is structural component of nitrogenase enzyme. This molybdenum is a structural component of nitrogenase enzyme. Before that, you see, I told you that plant take up molybdenum in MOO4 2- form, this molybdate ion. But in the plant system, under oxidative environment, it may exist as molybdenum 6. You write MO6 plus 6- sorry, and also undergoes reduction to MO5 also. So valency can take up this -2, -6, and -5, -4 also. 6, 5, 4, and 2 different valencies of molybdenum. Now coming to this nitrogenase enzyme. So molybdenum is a structural component of nitrogenase enzyme, and this enzyme has molybdenum iron protein. So if you have studied the biological nitrogen fixation, you must have heard that this nitrogenase enzyme is involved in biological nitrogen fixation. It is having two proteins actually. One is bigger protein, another is smaller one. So the bigger protein, that is iron molybdenum protein. We are talking about that only. So this molybdenum is a structural component of nitrogenase enzyme. This nitrogenase enzyme is having molybdenum iron protein and catalyze biological nitrogen fixation. That is why the molybdenum requirement of the nodulated legume is particularly high, because legumes require this nitrogenase enzyme to fix atmospheric nitrogen, right? So molybdenum is required for leguminated, this nodulated legumes. Another is nitrate reductase activity has been used as an indicator of status of molybdenum in plant. You see, one is that molybdenum is a component of nitrogenase enzyme. Another is that nitrate is reduced by nitrate reductase enzyme in the cytoplasm by the transfer of electron from molybdenum to nitrate. Nitrate reductase, what does it do? It reduces nitrate to nitrite form, right? And for reducing nitrate to nitrite form, it requires electron. And this electron is transported from molybdenum. So, owing to this close relationship between molybdenum supply, nitrate reductase activity, and the plant growth, this nitrate reductase activity has been used as an indicator of status of molybdenum in the plant. क्या बोला मैंने? नाइट्रेट रिडक्टेज का काम है नाइट्रेट को नाइट्राइट में रिड्यूस करना, और नाइट्रेट को नाइट्राइट में रिड्यूस करने के लिए इलेक्ट्रॉन की रिक्वायरमेंट होगी. वो इलेक्ट्रॉन ट्रांसफर किया जाएगा मोलिब्डेनम से, क्योंकि मोलिब्डेनम क्लोजली रिलेटेड है इस नाइट्रेट एक्टिविटी रिडक्टेज एक्टिविटी से, इसलिए नाइट्रेट रिडक्टेज एक्टिविटी हैज बीन यूज्ड एज एन इंडिकेटर ऑफ स्टेटस ऑफ मोलिब्डेनम इन द प्लांट्स. So you remember that molybdenum influences nitrogenase enzyme activity, nitrate reductase activity, and it affects the formation and viability of pollens and development of anthers. Critical concentration of molybdenum deficiency is less than 0.1 ppm, and since this molybdenum is affecting nitrogenase activity, nitrate reductase activity, so all in all it is affecting nitrogen cycle in the plant, in the soil, no? So nitrogen availability is influenced by molybdenum. So if molybdenum deficiency is there, there will be deficiency of nitrogen also. So they resemble each other. The most common symptom disorder of this molybdenum deficiency is whiptail in cauliflower, which is very frequently asked in exam. Whiptail of cauliflower due to molybdenum deficiency. Now moving ahead to another element, that is chlorine. So chlorine is absorbed as chloride ions by the plant, and chloride content in healthy plant is 100 to 500 ppm. You see, the incidences of chloride deficiency have not been reported in India. Why so? Chlorine की deficiency क्यों report नहीं है? Because of its large. हाँ बोलो.
Because मैंगनीज की भी इसके साथ associated होता है.
Okay, one of the reason is this only. Any other? Okay, you see, you have heard of this muriate of potash, no? KCl. One reason is this. Another is, you see, because of its large abundance in the air and its accretion through large number of fertilizers like MOP, ammonium chloride. So it is unintentionally being added to the soil. So it is sufficient for the plant. So deficiency of chloride is not reported in India because of its larger abundance in air. Another is because of its presence in the fertilizers like MOP and ammonium chloride. And it plays a major role in osmoregulation. It is a cofactor of manganese containing water splitting enzyme of photosystem 2. We have seen that manganese is associated with water splitting enzyme. So this chlorine is cofactor. So it also helps in this water splitting enzyme. Chloride in abundance suppresses the plant disease. So it also imparts disease resistance to the plant, improves the nutritional quality of the vegetables. And if it is less than 100 ppm, there will be deficiency. And deficiency symptoms of chlorine, they are similar to manganese deficiency. Now coming to factors affecting the availability of micronutrients in the soil. So you see, the effect of soil environment on the micronutrient cations are different from those of the micronutrient anions. Soil environment का effect means soil में कौन सा nutrient present है, क्या pH है, क्या redox condition है, they'll have different impact on cations and anions. So we'll discuss them separately. Okay. So first we'll discuss what is the effect of soil environment on cations. So cations कौन-कौन से हैं हमारे micronutrient cations? एक बार कोई बोल दो.
Iron, manganese, zinc, copper, nickel.
Yeah. 5 na? Okay, now we'll see the effect of soil pH on cations. You see, soil pH predominantly regulates the solubility and availability of micronutrient cations in the soil. How? The activities of micronutrient cations are inversely related to the pH. If the pH will be high, the activity of the micronutrient cation will be less. If pH is less, the activity will be more. Means in less pH range, the availability of the micronutrient cations is more. Why so? We'll see that. But yes, this is the information for you that activity of micronutrient cations is inversely related to pH. Activity of divalent and trivalent cations decrease 100 and 1000 fold with a unit increase in the soil pH. एक यूनिट पीएच के बढ़ने से, पहली बात तो हमें ये समझनी है कि पीएच के बढ़ने से माइक्रोन्यूट्रिएंट कैटायंस की एक्टिविटी कम हो जा रही है. अब कितनी कम हो रही है? एक यूनिट पीएच बढ़ने से डाइवेलेंट कैटायन की एक्टिविटी 100 टाइम कम हो जाती है, बिकॉज पीएच इज log 10 टू द log बेस 10 है ना, पीएच 10 बेस में चल रहा है तो वो 10 टू द पावर में हो जाएगा. सो 1 यूनिट इंक्रीज इन द soil pH will lead to decrease in trivalent cation and 1000-fold for trivalent cations. If it is monovalent, then there will be decrease in 10-fold. Okay, so you remember this one. Directly नहीं पूछेंगे, ऐसा पूछ लेंगे कि with each unit increase in the soil pH, what is the effect on activity of Fe2+? So it will decrease 100-fold because it is divalent. If it is asked about trivalent ferric form Fe3+, then it will decrease around 1000-fold. Micronutrient cations are most soluble and available under acid conditions. One thing we have seen कि ऐसा होता है, लेकिन क्यों हो रहा है? So micronutrient cations, they are more soluble and available under acid conditions because micronutrient cations are changed to hydroxy ions with increase in the pH. So with increase in the pH, these micronutrient cations, they are changed to hydroxy ions and get converted to insoluble hydroxides or oxides of the element. So pH बढ़ने की वजह से वहां पे हाइड्रोक्सिल आयन ज्यादा होंगे ना हायर pH पे, तो micronutrient cations, they are converted to insoluble hydroxides and oxides. That is why their availability is less at higher pH. And all the hydroxides of this micronutrient cations, they are sparingly soluble, but the degree of their solubility varies. ऐसा नहीं है कि सारे micronutrient cations के hydroxy ions जो भी बनेंगे hydroxides और oxides, वो किसी एक particular pH में जाकर ही dissolve होंगे. Their solubility varies, but yes, इतना sure है कि इनकी solubility कम होती है, जिसकी वजह से pH बढ़ने से micronutrient cations की availability कम हो गई. You see, the formation of insoluble hydroxide starts as soon as the pH of the soil solution is raised to 6 and above. अब ये हाइड्रोक्साइड्स कितने pH के बाद बनना शुरू होंगे? जैसे ही सॉइल का pH 6 या 6 से ऊपर जाएगा, ये हाइड्रोक्साइड्स और ऑक्साइड्स बनने लगेंगे, जिसकी वजह से माइक्रोन्यूट्रिएंट कैटायंस की अवेलेबिलिटी कम हो जाएगी. So the formation of insoluble hydroxide starts as soon as the pH of the soil solution is raised to 6 and above. So maintenance of soil pH around 6 for maintaining adequate concentration of these cations in the soil solution is necessary. So अगर हमें micronutrient cations की availability plant को पहुंचानी है, so the pH has to be less than 6, because at pH more than 6 there will be formation of insoluble hydroxides and oxides. This is one thing. I'll ask you one question after 2 slides, so you listen to me very carefully. Okay. Now another is, we'll see the effect of redox conditions on the availability of these micronutrient cations. So the lower oxidation states of cations means the reduced state. They are more soluble than the higher ones at the normal pH range of the soil. If you consider iron, we have Fe2 plus and we have Fe3 plus, ferrous and ferric form. Ferrous is reduced form, ferric is oxidized form, right? So the lower oxidation state of the cation means Fe2 plus. They are more soluble than the higher one, that means Fe3 plus. Okay, so Fe2 plus is more soluble than Fe3 plus. And under oxidized conditions, ऐसा क्यों हो रहा है? Under oxidized conditions, the oxides and hydroxides of the cations become more stable. See, reduced condition is more soluble than the oxidized form. Why? Because under oxidized condition, oxides and hydroxides of the cations, they are formed and they are more stable. Okay, so they're less soluble. And usually, hydroxides of the higher valent cations precipitate at lower pH than that of the lower valent cations. So जो भी पहली बात तो ये हो गई कि reduced condition में ज्यादा soluble होंगे cations. क्यों? क्योंकि जैसे ही oxidized condition आएगी, ये cations अपने oxides और hydroxides बना लेंगे, और hydroxides और oxides stable होते हैं, इसलिए soluble नहीं होंगे. दूसरी बात, usually hydroxides of higher valent cations यहाँ पे एक ही cation को लेना है, उसकी higher valency और lower valency को consider करना है. So hydroxides of higher valent cations means hydroxides of Fe3 plus, it precipitate at lower pH. Fe3 plus का hydroxide कम pH पे ही precipitate हो जाएगा, जबकि Fe2 plus का hydroxide अगर होगा भी तब भी वो lower pH पे इतनी जल्दी precipitate नहीं होगा. So it is more soluble. Making sense to all of you? Yes? Hello?
मैम एक बार रिपीट कर दो प्लीज.
ओके, पहली बात हमने ये बताई कि जो लोअर ऑक्सीडेशन स्टेट है कैटायन की, अगर हम आयरन की बात करें तो इट इज Fe2 plus. So Fe2 plus is more soluble than Fe3 plus. This is one thing. Why is it so? इसका explanation है आगे. क्योंकि ऑक्सीडाइज्ड कंडीशन में ऑक्साइड्स और हाइड्रोक्साइड्स बनेंगे ना. रिड्यूस्ड कंडीशन में तो वो Fe2 plus ही है, लेकिन अगर ऑक्सीडाइज्ड कंडीशन में है तो वहां पे ऑक्साइड्स और हाइड्रोक्साइड बनेंगे, फेरिक ऑक्साइड और फेरिक हाइड्रोक्साइड्स, which are more stable. तो उसकी अवेलेबिलिटी कम हो जाएगी, इसलिए रिड्यूस्ड फॉर्म जो है वो ज्यादा अवेलेबल है प्लांट को एज कंपेयर टू द ऑक्सीडाइज्ड फॉर्म. दूसरी बात, जो भी हमारा Fe2 plus है और Fe3 plus है, तो Fe3 plus का जो हाइड्रोक्साइड होगा और एक Fe2 plus का हाइड्रोक्साइड होगा, तो Fe3 plus का हाइड्रोक्साइड लोअर pH पे प्रेसिपिटेट हो जाएगा. इसीलिए एसिडिक रेंज में Fe3 plus की अवेलेबिलिटी प्लांट को कम होती है, Fe2 plus ज्यादा अवेलेबल है क्योंकि वो प्रेसिपिटेट नहीं कर रहा है. जबकि Fe3+ जो है वो प्रेसिपिटेट कर जा रहा है। मेकिंग सेंस नाउ? Yes?
Yes, yes, ma'am. Yes, ma'am.
Okay, another is you see the hydroxide of ferric precipitate, as I said कि जो भी हायर वैलेंट है उसका हाइड्रोक्साइड लोअर पीएच पे प्रेसिपिटेट हो जाएगा, लेकिन कितना लोअर, तो वैल्यू में भी देखते हैं। सो हाइड्रोक्साइड ऑफ Fe3+ precipitate in the pH range of 3 to 4. So 3 to 4 pH में फेरिक हाइड्रोक्साइड प्रेसिपिटेट हो जाएगा, whereas this ferrous hydroxide it requires a pH range of more than 6 for it to precipitate. हमें ये पता है कि हमारे माइक्रोन्यूट्रिएंट एसिडिक रेंज में अवेलेबल हैं, और एसिडिक रेंज में फेरस फॉर्म प्रेसिपिटेट नहीं होता है, इसलिए प्लांट यूज टू टेक दिस फेरस फॉर्म rather than this ferric form because it used to precipitate at pH 3 to 4. So this is one of the reason why the availability of iron is poor in soil once it is converted to ferric form. So availability of iron, if you see, this ferrous form is available to the plant rather than this ferric form, because this ferric form will precipitate at pH 3 to 4, whereas this ferrous form require pH more than 6 to precipitate. So it will not precipitate in the acidic range. Making sense to all of you?
Yes, ma'am.
So दो बातें हमने पढ़ी, पहला pH का इफेक्ट, सो एसिडिक पीएच में माइक्रोन्यूट्रिएंट कैटायंस अवेलेबल ज्यादा होंगे, हायर पीएच पे नहीं होंगे। रिडॉक्स की बात करें तो रिड्यूस्ड फॉर्म प्लांट को ज्यादा अवेलेबल होगा एज कंपेयर टू द ऑक्सीडाइज्ड फॉर्म। दीज आर द 2 पॉइंट्स दैट वी हैव डिस्कस्ड। नाउ कम्स द क्वेश्चन फॉर यू, यू थिंक एंड यू टेल। Question is, what is the effect of submergence of acid soil on the micronutrient cations availability? किन किन बातों पे ध्यान देना है? पहली बात submergence यानी water logged condition है, इसको रिलेट करो redox से। दूसरी बात एसिडिक सॉइल है, ये भी ध्यान रखना है। और तीसरी बात हम सिर्फ कैटायंस की बात कर रहे हैं, ठीक है? तो थोड़ा सोचो और मुझे बताओ कि चलो एक एक करके इसको ब्रेक करते हैं क्वेश्चन को थोड़ा आसान करने के लिए। What is the effect of submergence on कैटायंस अवेलेबिलिटी? सॉइल हमारी एसिडिक है, तुम मुझे ये बताओ कि एसिडिक सॉइल में सबमर्जेंस का इफेक्ट क्या होगा? व्हाट इज द इफेक्ट ऑफ सबमर्जेंस ऑन पीएच ऑफ एसिडिक सॉइल, वेदर इट विल इंक्रीज और डिक्रीज?
मैम वो न्यूट्रल
मैम
इंक्रीज
होगी मैम,
इंक्रीज होगा। तो अगर एसिडिक सॉइल है, सबमर्जेंस की वजह से पीएच इंक्रीज होगा। अगर पीएच इंक्रीज हो रहा है तो माइक्रोन्यूट्रिएंट अवेलेबिलिटी कम होगी, ज्यादा होगी?
कम हो जाएगी।
कम हो जाएगी।
हाँ, सबको ये बात समझ आ रही है ना? पहली बात कि सबमर्जेंस अगर एसिडिक सॉइल का हो रहा है तो माइक्रोन्यूट्रिएंट अवेलेबिलिटी कम हो जाएगी क्योंकि पीएच बढ़ रहा है। इतनी बात सबको समझ आ रही है?
यस मैम।
हाँ, अगली बात मैंने ये बताया कि जो रिडॉक्स फॉर्म, रिड्यूस्ड फॉर्म होती है वो प्लांट को ज्यादा अवेलेबल होती है एज कंपेयर टू द ऑक्सीडाइज्ड फॉर्म। तो सबमर्जेंस की वजह से रिड्यूस्ड फॉर्म क्रिएट हो रही है या ऑक्सीडाइज्ड? रिड्यूस्ड फॉर्म। तो प्लांट की को तो अवेलेबिलिटी बढ़ जानी चाहिए।
ऑक्सीडाइज्ड।
मेरी बात समझ पा रहे हो मैं क्या बताना चाह रही हूँ?
मैम रिड्यूस्ड फॉर्म Fe2+ बाय Fe3+।
हाँ, किसी भी कैटायन की बात कर लो। पहली बात मैंने ये बताई। हाँ
मैम
बोलो।
अगर एग्जांपल लेते हैं तो Fe2+ या Fe3+, किसका होगा इसमें?
रिड्यूस्ड फॉर्म Fe2+ है।
Fe2+ का ज्यादा हो।
हाँ, यू सी, ठीक है, मैं दोबारा से बोलती हूँ तो मेरी बात ध्यान से और समझेंगे। अभी शायद मेरा क्वेश्चन नहीं समझ आ रहा है। सवाल ये है कि एसिडिक सॉइल है और सबमर्जेंस कर रहे हैं, तो क्या माइक्रोन्यूट्रिएंट कैटायंस की अवेलेबिलिटी बढ़ जाएगी या घट जाएगी? ये सवाल है, ठीक है। अब सबमर्जेंस की वजह से एसिडिक सॉइल का पीएच टुवर्ड्स न्यूट्रल जाएगा, यानी कि अब एसिडिक रेंज से पीएच बढ़ गया है। क्योंकि पीएच बढ़ गया है तो माइक्रोन्यूट्रिएंट कैटायंस की अवेलेबिलिटी कम हो जानी चाहिए, पहली बात। दूसरी बात मैंने ये बताई कि जो रिड्यूस्ड फॉर्म होती है वो ज्यादा अवेलेबल होती है एज कंपेयर टू द ऑक्सीडाइज्ड फॉर्म। तो अगर सबमर्जेंस कर रहे हैं यानी कि हम रिड्यूस्ड कंडीशन क्रिएट कर रहे हैं, तो अगर रिड्यूस्ड कंडीशन क्रिएट हो रही है तो मतलब माइक्रोन्यूट्रिएंट भी तो रिड्यूस्ड फॉर्म में होगा, तो उसकी अवेलेबिलिटी तो बढ़ जानी चाहिए। पहली स्टेटमेंट में हम बोल रहे हैं कि सबमर्जेंस की वजह से माइक्रोन्यूट्रिएंट कैटायंस कम हो रहे हैं। दूसरी बात हम बोल रहे हैं कि सबमर्जेंस की वजह से माइक्रोन्यूट्रिएंट कैटायंस बढ़ जाने चाहिए। दोनों कॉन्ट्राडिक्टरी बातें हैं, क्या मेरी बात समझ पा रहे हो?
यस मैम, बिकॉज ऑफ रिड्यूस कंडीशन बढ़ जानी चाहिए।
दोनों कॉन्ट्राडिक्टरी बात है, लेकिन फिर नेट इफेक्ट क्या होगा? सबमर्जेंस की वजह से माइक्रोन्यूट्रिएंट कैटायंस बढ़ेंगे या घटेंगे, ये हम कैसे बताएं? तो अब मैं बोल रही हूँ, धीरे धीरे बोलूंगी, मेरी बात ध्यान से सुनना, ठीक है? यू सी, इन जनरल, if acid soil is brought under prolonged submergence. The pH gradually approaches neutrality, leading to the reduction in the solubility of the micronutrient cations. On the other hand, dissolution of iron, manganese oxide enhances the solubility and availability of micronutrient cations with the reduction of redox potential. पहली बात में हमने बोला कि pH बढ़ने से माइक्रोन्यूट्रिएंट कैटायंस कम हो जाने चाहिए. दूसरी बात हम बोल रहे हैं रिड्यूस्ड कंडीशन होने की वजह से माइक्रोन्यूट्रिएंट कैटायंस बढ़ जाने चाहिए. अब ये कॉन्ट्राडिक्टरी बातें हैं. Under such condition, the net effect of these two contrasting phenomena on the availability of micronutrient cations depends on the initial soil pH and the period under submergence. Initial soil का pH कितना है? बहुत ज्यादा एसिडिक है और सबमर्जेंस की वजह से हो सकता है वो 6 के ऊपर ना पहुंचा हो, तो माइक्रोन्यूट्रिएंट अवेलेबल हो सकते हैं. अगर इनिशियली उसका पीएच 4 या 5 है और हम सबमर्जेंस करेंगे तो वो 6 से ऊपर चला ही जाएगा, तब वहां पे माइक्रोन्यूट्रिएंट डेफिशिएंसी हो जाएगी. तो इनिशियल सॉइल पीएच पे डिपेंड करेगा सबमर्जेंस का इफेक्ट. दूसरी बात, कितने टाइम के लिए सबमर्जेंस कर रहे हो, मतलब रिड्यूसिंग कंडीशन कितनी ज्यादा है, इस पे भी डिपेंड करेगा कि माइक्रोन्यूट्रिएंट अवेलेबिलिटी कितनी है. Is it making sense to all of you?
Yes, ma'am.
Okay. Can I move ahead or anyone want me to explain it again?
Yes, ma'am.
ठीक है, चलते हैं आगे. Okay, now moving ahead. अच्छा, अब सोचो कि ये तो एसिडिक सॉइल की बात थी. If the soil is alkaline, what is the effect of submergence on micronutrient cation availability? इसी तरह से सोचो कि अगर अल्कलाइन सॉइल है, सबमर्जेंस करेंगे तो पीएच बढ़ेगा घटेगा?
पीएच घटेगा मैम.
घटेगा. और पीएच कम होने की वजह से कैटायंस की अवेलेबिलिटी बढ़ जाएगी या कम हो जाएगी?
बढ़ जाएगी.
बढ़ जाएगी. हाँ, तो अल्कलाइन सॉइल है तो वहां पे सबमर्जेंस की वजह से माइक्रोन्यूट्रिएंट कैटायंस की अवेलेबिलिटी बढ़ जाएगी. और अब रिड्यूस्ड कंडीशन का भी इफेक्ट देख लो. अगर सबमर्जेंस कंडीशन है तो रिड्यूस्ड कंडीशन होगी तो माइक्रोन्यूट्रिएंट कैटायंस इजीली अवेलेबल हो पाएंगे. सो एसिडिक सॉइल में ये कॉन्ट्रास्टिंग बात है कि पीएच और रिडॉक्स का इफेक्ट, लेकिन अल्कलाइन सॉइल में पीएच भी कम आएगा 6 से नीचे और रिड्यूस्ड कंडीशन भी होगी. सो दोनों ही केस फेवरेबल हैं फॉर माइक्रोन्यूट्रिएंट कैटायन अवेलेबिलिटी. Okay, so whenever this kind of question is asked to you, you focus on whether it is asking about acid soil or alkaline soil. Okay, now let us move ahead to another factor that affects the availability of micronutrient cations. So it is soil constituent. You see, the availability of micronutrient cations in the soil is highly affected by solid phase such as silicate clays, oxides of aluminum, iron or manganese, organic matter content. So they all influence the availability of micronutrient cations. How? Micronutrient cations may be tightly bound or fixed in the octahedral layer, especially in 2:1 type of clay minerals. In 2:1 type of clay minerals, there may be chances that these micronutrient cations may be tightly bound or fixed in the octahedral layer. So it will not be available to the plant. And such a fixation of micronutrient by the soil clay, they may cause serious problem for zinc and copper especially. You remember that fixation of zinc and copper is mostly in octahedral layer of 2:1 type of the clay minerals. For iron and manganese, even if it is fixed, but their deficiency does not occur because they are already in high amount in the soil. ठीक है, आयरन मैग्नीज ऑलरेडी ज्यादा अमाउंट में है सॉइल में, अगर कुछ अमाउंट फिक्स भी हो जाता है तो भी प्लांट को बाकी अवेलेबल हो जाएगा. But this fixation is a major problem with zinc and copper. Okay. Another is this cations can form both soluble and insoluble compounds with organic matter. Cations, they can form both soluble and insoluble compounds with the organic matter besides being incorporated in it as a structural component. During the decomposition of organic matter, if these cations are adsorbed or fixed or they have formed complex or compounds with the organic matter, so when there is decomposition of organic matter, these nutrient cations are slowly released into the soil, which can be easily taken up by the plant. But the problem occurs when the complexes are insoluble. अगर ये complex जो micronutrient ऑर्गेनिक मैटर से बना रहे हैं, अगर वो सॉल्युबल है तो अच्छा ही है कि उसका लॉसेस कम होगा, ऑर्गेनिक मैटर के साथ कॉम्प्लेक्स बन के धीरे धीरे रिलीज होता रहेगा और प्लांट ले लेगा. लेकिन अगर ये कॉम्प्लेक्स इनसॉल्युबल है तब वहां पे प्रॉब्लम है. बट So the cationic micronutrients, they can form organometallic complexes, that is chelates with the organic molecules. And if that particular chelate is soluble, then it is actually good because it is preventing the losses, right? But the formation of insoluble complexes, they make these nutrients unavailable. And this is the major problem with copper. Copper is having very high affinity for this organic matter. So once it forms bonds with the organic matter, it forms insoluble compound. That is why reclamation disease we have started. So copper is retained very strongly in the organic soil. बाकी सारे एलिमेंट भी रिटेन होते हैं ऑर्गेनिक मैटर से, लेकिन वो सॉल्युबल कंपाउंड बनाते हैं जो कि धीरे धीरे रिलीज होकर प्लांट को मिल जाता है. लेकिन कोई एलिमेंट अगर बहुत टाइटली कॉम्प्लेक्स बना ले जो कि प्लांट को अवेलेबल ना हो तो उसकी डेफिशिएंसी हो जाएगी, राइट? So this is how this organic matter influence the availability of cations. Now moving ahead to the effect of these factors on the availability of anions. So among the three anions that we have studied, chloride, three anions that we have studied, that is chloride and molybdenum, and another is which one? Boron. So among these three anions, chloride is absorbed in large quantity by the plants and its availability in soil is not affected by any soil property. So ऐसा हमने देखा भी कि chloride deficiency report भी नहीं हुई है because it is actually available in large quantity. So its availability is not affected by any soil property as most of the chloride compounds are water soluble. Another is soil texture appears to be the most important factor governing the boron availability. Texture is the most important factor that governs the boron availability. In general, sandy soil contain less available boron than heavy soil. Sandy soil, this heavy soil is clay soil. So sandy soil contain less available boron. Why? Because boron is present as boric acid, which can be leached down from the light textured soil. So boron is present in boric acid form. In light textured soil, means in sandy soil, it gets leached down. But in case of heavy soil, clay, it is actually retained. So the loss of boron is more in sandy soil. So it is less available in sandy soil because of its leaching. And boron is more tightly absorbed on humus than on inorganic colloids. So organic matter is practically the main source of boron in many soils. So boron is absorbed on the humus, and as soon as the decomposition of this organic matter occurs, there is availability of boron. Now, why boron deficiency occurs under drought conditions? I am telling you that under drought conditions, there is deficiency of boron. Now you tell, I have told you the reason already. In सारे points में इसका reason है, तो कोई मुझे बताएगा why there is deficiency of boron under drought condition?
Ma'am, to reduce mineralization.
Yeah.
Under drought conditions.
Mineralization of what? Okay, you see, I told you that organic matter is practically the main source of boron in many soils because boron is more tightly absorbed in the humus, right? So under drought condition, boron deficiency in the plant is observed due to reduction in organic matter, mineralization and uptake by the plant. इसलिए drought condition में boron की availability कम होगी क्योंकि organic matter का decomposition नहीं हो पाएगा. Another is molybdenum is the only essential element whose availability consistently increase with increase in the pH. pH बढ़ने से molybdenum एक अकेला element है जिसकी availability बढ़ती ही है, ठीक है? And high content of iron and ammonium oxide in the soil reduces the availability of molybdenum. Why molybdenum is less available in the acidic range? Because of this iron aluminum oxide. So molybdenum gets fixed in this iron aluminum oxides as well as on the silicates at low pH through ligand exchange. That is why this availability of boron is less. Molybdenum is less under acidic range and it is high under at high pH. Okay. Now coming to soil testing method for this micronutrients. So I'll tell you a little bit brief about this DTPA, the process, the functions, and the principle on which DTPA actually works. So you tell me what is the full form of DTPA? Yes, anyone?
Diethylene triamine pentacetic acid.
Yeah, good. So it is diethylene triamine pentacetic acid. This is an extracting reagent for micronutrients. right? So DTPA is an extracting reagent given by Lindsay and Norvell in 1978. So DTPA is given by Lindsay and Norvell, very frequently asked, 1978. And the reagent, it is not only DTPA, there are two other reagent along with DTPA. So reagent you write, this is frequently asked in exam. You write 0.005 molar DTPA, 0.005 molar DTPA plus 0.01 molar calcium chloride dihydride, 0.01 molar calcium chloride dihydride plus 0.1 molar T triethanol amine. So I'm repeating 0.005 molar DTPA plus 0.01 molar calcium chloride dihydride plus 0.1 molar TEA triethanol amine. Written all of you? कोई एक भी रहता है तो मैं लिख के भी बता सकती हूँ. Yes.
हो गया मैम.
Okay. Now the pH at which this DTPA works, it is 7.3. Okay, so Lindsay Norvell has given this reagent. I told you it is DTPA, calcium chloride, and T. pH is 7.3. Okay, very frequently asked question. Now we'll see the principle on which this DTPA works. Okay. For that, there is an option of whiteboard. Where is it?
Ma'am, you take a blank page, then you take the pencil, ma'am. Yes.
अभी ये visible है क्या whiteboard?
Yes, ma'am. It's visible.
हाँ, ठीक है. Okay, so I'll tell you the principle on which DTPA works. Okay, so you see, we know that there is solid phase. There is solid phase in soil, right? And there is solution phase. Plant take up nutrients from which phase? Directly from solution, na?
Solution.
So plant take up nutrients. This is our plant, हाँ? ऐसे सोच लेते हैं, ये हमारा plant है. So this is our plant. It takes nutrient from solution, and if from solution, soil solution, particular nutrient is taken up by the plant, then it is replenished from solid phase. अगर plant यहाँ से ले लेगा nutrient, तो यहाँ पे कहाँ से आएगा? Solid phase से ही आएगा. और अगर हमने कोई फर्टिलाइजर डाला, सोल्यूशन वाले में डाला, अगर ये ज्यादा है तो वो सॉलिड फेज में चला जाएगा. इतनी बात सब समझते हैं?
Yes, ma'am.
Yes. So this solid phase is actually replenishing whatever nutrient is taken up by the plant from the solution phase. This is one thing. Now you see, if we are adding DTPA in soil solution, हम DTPA डाल रहे हैं. What is this DTPA? DTPA is a chelating agent. Chelating agent means जो किसी भी nutrient को bind कर ले. जैसे कि एक पक्षी का पंजा होता है. Chelate terms comes from the claw word, which means बस उसको बांध लेना है एकदम. Okay, complex बनाना है, chelate करना है. So this DTPA is a chelating agent. If we add this DTPA in the soil solution, what this DTPA will do? This will bind the micronutrient. है ना? This DTPA will chelate the micronutrient. Okay. And if this DTPA will chelate this micronutrient, then the concentration of micronutrient in solution will be less. तो वो कहाँ से आएगा? अगर यहाँ पे solution में कम है तो किधर से आएगा nutrient? Solid phase से?
Solid phase.
है ना? So one thing is, if plant take up this micronutrient from the soil solution, it will replenish by the solid phase. This is one. Another is, if we are adding DTPA, this DTPA will bind the micronutrient, and the availability of my particular micronutrient to plant will not be there, and it will be replenished by the solid phase. Okay. So जो principal DTPA जिस पे काम करता है वो ये कहता है that the chelated micronutrient, जो micronutrient इस DTPA ने chilate किया है, it will lose its ionic form as if absorbed by the plant roots. जो micronutrient DTPA ने chilate किया है वो ऐसे behave करेगा जैसे कि plant ने उसे uptake कर लिया है. क्योंकि solid phase को तो नहीं पता ना कि plant ने लिया है या DTPA ने bind किया. उसको तो सिर्फ इतना मतलब है कि solution से कम हो गया, है ना? So DTPA works on the principle that whatever the nutrient is chelated by the DTPA, it will mimic as if it is taken up by the plant. और अगर ये प्लांट ने ले लिया, ऐसा मिमिक कर रहा है, तो सॉलिड फेज भी माइक्रोन्यूट्रिएंट रिलीज कर देता है सोल्यूशन में. अब हमारे पास जो भी सोल्यूशन में माइक्रोन्यूट्रिएंट है वो डीटीपीए ने चिलेट करा और उसको हम पता कर सकते हैं. ठीक है, तो प्रिंसिपल क्या है, अगर सोल्यूशन में न्यूट्रिएंट कम होगा तो वो सॉलिड फेज से आ जाएगा. ठीक है, और सोल्यूशन में कम कैसे होगा, जब प्लांट उसको अपटेक करेगा. तो इफ प्लांट इज टेकिंग न्यूट्रिएंट फ्रॉम द सॉइल सोल्यूशन, इट विल बी रिप्लेनिश्ड बाय द सॉलिड फेज. और डीटीपीए डालने की वजह से वो उस पर्टिकुलर माइक्रोन्यूट्रिएंट को चिलेट कर देता है, उसको बाइंड कर देता है और ऐसे बिहेव करता है जैसे कि वो न्यूट्रिएंट प्लांट ने ले लिया. और अगर सॉलिड फेज को लग रहा है कि सोल्यूशन में कम हो गया है, तो वो वापस से सोल्यूशन में देगा और फिर से डीटीपीए उसको बाइंड कर लेगा और डीटीपीए हमें बता देगा कि कितना न्यूट्रिएंट सॉइल में है. इज इट मेकिंग सेंस टू ऑल ऑफ यू?
यस मैम. मैम तो मेजर, मैम अल्टीमेटली इसको मेजर कैसे करेंगे मैम?
डीटीपीए ये तो डबल एस से रीडिंग लेंगे ना. डीटीपीए ने जो भी न्यूट्रिएंट है उसको बाइंड करना है बस, और डीटीपीए ने जो भी बाइंड करा वो डबल एस से रीडिंग ले लेंगे. ठीक है, डीटीपीए इज ओनली एन एक्सट्रैक्टिंग एजेंट, जो सॉल्यूशन में जो भी है उसमें से माइक्रोन्यूट्रिएंट को निकाल ले बस, और बाकी रीडिंग तो डबल एस में लेना है ना, एटॉमिक एब्जॉर्प्शन में. मेकिंग सेंस?
तो मैम एक्चुअली हम लोग ये पता कर रहे हैं कि सॉइल में कौन कौन सा माइक्रोन्यूट्रिएंट है,
कितना है, कौन है और कितना है. क्या कितना अमाउंट भी तो पता कर रहे हैं ना. और एक बात और, इस ये बताने का पर्पस ये था कि तुम दो बात समझते हो, इंटेंसिटी फैक्टर और क्वांटिटी फैक्टर.
यस मैम.
हाँ, क्या होता है बताओ.
Intensity is the concentration of a particular nutrient in the soil solution, and there is the ability of the soil to maintain that concentration. मतलब how to replenish that.
तो हम कह सकते हैं क्वांटिटी फैक्टर इज व्हाटएवर इज देयर इन द सॉलिड फेज इज क्वांटिटी, इंटेंसिटी फैक्टर इज दिस सॉल्यूशन. प्लीज म्यूट योर माइक. बस जितने टाइम बोलना हो उतना बोलो, फिर म्यूट कर दो. ठीक है. So this quantity factor is whatever there is in solid phase and intensity is there in solution phase. और मैंने बोला कि डीटीपीए ने पहले जो सोल्यूशन में उसको तो बाइंड कर लिया, फिर ऐसा मिमिक किया कि वो प्लांट ने ले लिया है और फिर सॉलिड फेज ने भी इधर दे दिया. So DTPA is actually estimating both intensity and quantity factor. It gives not only initial concentration but also quantity factor related to labile pool. It takes into account both intensity and quantity factor. Is it making sense to all of you? Yes?
मैम रिपीट एक बार, वन सेकंड, लास्ट लाइन.
हाँ, ठीक है. तुम मुझे बताओ अभी तक ये प्रिंसिपल में तुमको क्या समझ आया? पहले व्हाट्स योर गुड नेम?
मैम ऋतिक कुमार.
हाँ ऋतिक, यू से.
मैम जैसे डीटीपी एक चिलेटिंग एजेंट है, इसको हम सॉइल सॉल्यूशन में डालते हैं, तो वहां से वो माइक्रोन्यूट्रिएंट को ऑब्जर्व कर लेता है. तो जो भी सॉइल सॉल्यूशन में चिलेट कर लेता है, तो जो भी सॉइल सॉल्यूशन माइक्रोन्यूट्रिएंट है उसको रिप्लेनिश करने के लिए सॉइल से माइक्रोन्यूट्रिएंट आता है वापस, और फिर इसे हम ऑब्जर्व कर सकते हैं. तो इससे पता चलता है कितना माइक्रोन्यूट्रिएंट सॉइल में ऑब्जर्व हुआ, मतलब डीटीपीएस ऑब्जर्व हुआ.
ठीक है, बिल्कुल सही बोल रहे हो. बस एक बात, जब भी ये प्रिंसिपल बोलोगे तो ये बात भी बोलना कि डीटीपीए इज चिलेटिंग माइक्रोन्यूट्रिएंट एंड बिहेविंग एज इफ इट इज टेकन अप बाय द प्लांट. तभी तो सॉलिड फेज से रिप्लेनिश हो रहा है ना, क्योंकि ये मिमिक कर रहा है ऐसा कि वो प्लांट ने ले लिया. ठीक है, अब तुमने खुद ही बोला बात अपनी कि एक तो जो सॉल्यूशन में है वो डीटीपीए ने लिया और उसकी वजह से सॉलिड फेज से जो आया वो भी हमने मेजर कर लिया. So DTPA method is actually estimating both intensity and quantity factor. This is what my last line was. Making sense now?
And quantity is on solid.
Quantity solid phase वाले को बोलते हैं जो कि replenish कर सकता है, जो labile pool है, जो replenish करके solution में आएगा. तो solid phase वाला भी तो solution में आया, तभी तो हमने उसे भी measure किया. ठीक है?
Yes ma'am.
है ना? तो DTPA measures both intensity and quantity factor. Any confusion? Any one of you? Yes or no? बोलना है, चुप रहने से तो मुझे नहीं पता चलेगा. Can I move ahead? All of you?
Yes ma'am.
Okay. So we have seen, हाँ बोलो, कोई कुछ कहना चाहता है? इसको फिर मैं डिलीट करूंगी ना, तब इसलिए पहले बोलो, ठीक है, वो कर देते हैं.
No, ma'am, please go ahead, ma'am.
हाँ, okay, fine. So we have seen that it is given by Lindsay and Orwell. We have seen the reagent, we have seen the principle, we have seen the pH, that is 7.3. Now another is complexing power of DTPA, as this DTPA is the extractant for all the micronutrients, no? So how much micronutrient this particular DTPA is able to complex, right? So you write the complexing power of DTPA is 10 times, 10 times its atomic mass. So whatever micronutrient we are taking into account, suppose if it is iron, so iron is having atomic mass 55. So 550 ppm of iron can be complexed with DTPA. Okay, so the complexing power of DTPA is 10 times its atomic mass. So iron atomic mass is 55. So 550 ppm iron can be complexed with DTPA. Manganese atomic mass is 56. So 560 ppm of manganese can be complexed. Zinc atomic mass is 66. So 660 ppm of zinc can be complexed with DTPA. Copper atomic mass is 63. So 630 ppm of copper can be complexed. So DTPA is able to complex 10 times the atomic mass. Okay, now moving ahead. Soil test for micronutrient. One of the extractant is DTPA that we have seen. There are other extractants also. DTPA is common for all, but yes, there are other extractants also. Like for zinc, HCl. Bolo.
Slides are not visible.
Okay, wait. अच्छा, व्हाइट बोर्ड अनशेयर किया तभी. अभी विजिबल है?
Yes, ma'am.
Okay. Yes, ma'am. So in this, I told you the complexing power. Now moving ahead to another soil testing extractants for different micronutrients. You see, DTPA is common for all the micronutrients, but yes, other than DTPA, there are other extractants also. Like for zinc, there is HCl, ammonium acetate, different concentration of HCl also. For copper, there is HCl, ammonium acetate. Iron, there is another one. For manganese, for nickel, for boron, molybdenum. What is important that I want to tell you in this is, one thing is for DTPA, you remember that the soil solution ratio is 1:2. Okay, whichever micronutrient you take, whether zinc, copper, iron, manganese, nickel, the soil solution ratio is 1:2. Okay, 1:2. I have highlighted it also. And it is shaken for 2 hours. DTPA को तो कम से कम हमें पूरा मेथड ध्यान रखना है. 2 hours shaking, soil solution ratio 1:2, pH is 7.3, different different chemical reagents we have seen, and temperature is 25 degrees Celsius. You're right, temperature is 25 degrees Celsius, shaking time 2 hours, 1:2 ratio. This is one thing. Another is, the question that is usually asked is critical level of deficiency. If we extract zinc by DTPA method, and if it is less than 0.5, then it is deficient. Okay, so this critical level is important from exam point of view. This critical level of these nutrients by DTPA method at least. बाकी method को फिर भी छोड़ सकते हो, लेकिन DTPA को देखेंगे, ठीक है? Another question that is usually asked is, this boron is estimated by simple hot water. It is by boiling hot water. Who has given this hot water method of boron?
Burger and
Yeah, Berger and
Trong.
Trog. So it is by Berger and Trog, this boiling hot water method of boron that is usually asked. Another is molybdenum. This molybdenum is extracted with ammonium acetate. What is this reagent known as? Ammonium oxalate. Any one of you? Hint for you, it starts from G.
Griggs reagent.
Yeah, wonderful. So molybdenum is estimated by ammonium oxalate, which is Griggs reagent, and the pH, you remember, it is frequently asked, it is 3.3. So Griggs reagent is having pH 3.3, and molybdenum extracted with ammonium oxalate, Griggs reagent. Boron, you remember, boiling hot water, Berger and Trog. Nickel, DTPA only. Manganese, iron, copper, zinc, DTPA only. Okay, now let us move ahead. There are different fertilizers. हाँ जी, yes.
मैम जैसे हम DTPA के साथ TEA भी यूज करते हैं ना, ट्राइथेनोल अमीन जैसे, मैम उसका क्या पर्पस है? लाइक अगर DTPA एक्सट्रैक्ट करके हमें एग्जैक्ट अमाउंट बता देता है, तो व्हाट्स द पर्पस ऑफ एडिंग TEA?
Okay, if you are asking about TEA, let me tell you about calcium chloride also. Means I'll tell you why calcium chloride is added and why TE is added. You see, this T is triethanol amine, right? So the purpose of adding T is that it clearly burnt in the flame of AAS. When we add TEA, then the flame in AAS burnt clearly. This is one of the reasons of adding T. Another is, if flame is not disturbed when TEA is added, as no residue is produced. If we'll not add T, so there will be production of residues. So there will be some choking in the flame of AAS. For that very matter, we add T. This is one. Making sense to you? Okay, right? Now coming to calcium chloride, why we are adding calcium chloride in it? So initially, when Lindsay and Norwell gave this method, gave this DTPA method, they gave it for calcareous soil. Initially, they gave it for calcareous soil, and in calcareous soil, calcium carbonate is there. So the challenge was to check the excess dissolution of calcium carbonate. So calcareous soil के लिए initially DTPA method बनाया. प्रॉब्लम ये थी कि जब DTPA डाल रहे थे तो जो सॉइल का कैल्केरियस सॉइल का कैल्शियम कार्बोनेट है वो सोल्यूशन में आ जा रहा था. तो सोल्यूशन में अगर हम पहले से ही कैल्शियम का कोई सोर्स डाल के रखेंगे तो सॉलिड फेज से कैल्शियम नहीं आएगा. So that is why this calcium chloride was added. Making sense to all of you?
Yes, ma'am. Due to concentration gradient.
Yeah, it was making actually, ऐसा बोल सकते हैं कि it was to check the excess dissolution of calcium carbonate. This was one of the reason of adding calcium chloride. Another is, we know that calcium helps in coagulation. It is a flocculating agent. So calcium helps in coagulation. So to precipitate the filtration for getting clear extract. अगर हम कैल्शियम का कोई सोर्स डालते हैं तो वो बाकी सब चीजों को फ्लोकुलेट कर लेगा और हमें जो एक्सट्रैक्ट मिलेगा वो क्लियर मिलेगा. और अगर क्लियर एक्सट्रैक्ट मिलता है तो AAS में चोकिंग नहीं होगी. सो हर चीज का रीजन है क्यों कितना क्या डाला. ठीक है. ओके, कैन आई मूव अहेड नाउ? यस.
Yes, ma'am.
So we were on fertilizers, different fertilizers of micronutrients. Their percentage can be asked in exam. Okay, boron, we used to add boric acid usually 17. This soluble sodium tetraborate. Potassium chloride, 47% chloride is there. For copper, copper sulfate, copper chelates, means copper EDTA is added. For iron, ferrous sulfate, ferric sulfate is added. Manganese, sulfate form is usually added of all the fertilizers. Okay. Commonly asked question is that zinc deficiency, since they are very much concerned with zinc deficiency. So they used to ask, what is the nutrient content in zinc sulfate, tetrahydrate and monohydrate? This zinc sulfate, it comes in two forms. This one is monohydrate. Another is zinc sulfate, heptahydrate. Sorry, I said tetra, that was my mistake. It is 7H2O. So ZnSO4, monohydrate and heptahydrate. Okay. So they used to ask, what is the content of zinc in monohydrate and heptahydrate? So you should know both. Okay. So in monohydrate, it is 36. In heptahydrate, how much? You tell.
21.
Somewhere it is written 21, somewhere 27 also. So it is in the range of 22, 25 or something. Okay. एक और बात, अगर हम सारे फर्टिलाइजर का न्यूट्रिएंट परसेंटेज याद कर सकते हैं, वेल एंड गुड. नहीं कर सकते तो कभी भी अगर पूछा जाए कि हमें बोल रहे हैं कि जो हमने 4 ऑप्शंस दिए हैं उसमें से किस पर्टिकुलर फर्टिलाइजर में हाईएस्ट कंटेंट है उस न्यूट्रिएंट का. सपोज आयरन का पूछ रहे हैं और हमारे पास ऑप्शन आयरन सल्फेट है, आयरन ऑक्साइड है और फेरिक सल्फेट, फेरस सल्फेट, EDTA, ये सब है. तो अगर ऑब्जर्व करेंगे हम तो जो ऑक्साइड फॉर्म है उसमें सबसे ज्यादा कंटेंट है. अगर ऐसा डायरेक्टली कभी आंसर करना है तो कर सकते हैं कि ऑक्साइड फॉर्म में जैसे अगर जिंक का भी देखें, यू सी द जिंक ऑक्साइड इज हैविंग हाईएस्ट nutrient content. Any other oxide form यहाँ दिया नहीं है, but if you see this in book, you will find that oxide form is having highest nutrient content. Okay, so that's all from my side. If you have anything to ask, if you want me to repeat anything, you can ask.
Can you please provide this recorded classes? Previous recorded classes.
I will provide this PPT to you. I don't know about the recording.
Ma'am,
PPT I will provide.
I didn't. I'm not
Recording
in group,
they'll also provide.
I'm not in group, ma'am.
Okay, whosoever is in group, who will take charge of all this? Sir, are you there? Suman sir?
Yes, I am here, ma'am. Yes.
So Suman sir,
I will add these boys in the group. I hope.
That's fine.
Okay, don't worry. You take the class.
Okay.
Anything else anyone want to ask, or any confusion, any clarification required?
Please ask the question. Please ask. If any question you have regarding to micronutrient, you ask to them.
Yeah, one more thing, this micronutrient chapter, you read from both ISSS book. That would be sufficient. This new ISSS book, Soil Science and Introduction and Fundamental of Soil Science, very wonderfully written in both the books. So that will be sufficient for you. लेकिन उसके बाद भी कोई specific query है, so you can prefer Tested and Nielsen. Otherwise, ISSS, both the books are sufficient. किसी को सिर्फ एक ही बुक पढ़ने का मन है, तो वो सॉइल साइंस एंड इंट्रोडक्शन जो न्यू ISSS है उसको प्रेफर करना है इस चैप्टर के लिए. एनीथिंग एल्स?
Hello, ma'am. My one question to you, ma'am.
Yes.
What is the dilution effect of the zinc?
What's your good name?
Myself, Suman, ma'am. Suman.
Yes, sir.
Yes.
Your question.
This is the question that is asked in the nitrogen chapter by the one student. I hope that there is a note percent here that class. I try to explain him, but I also request to you. You also explain these things to the student. Tamanna Sharma. I hope they asked a question in personally to my call.
Yes. What is the question?
Question, ma'am. What is the dilution effect of the zinc? Zinc. Dilution effect of zinc.
Okay. And what was the another question?
And ma'am, another question is tell something about what is the transportation of macronutrient to the plant root.
Okay.
And a brief idea about the transportation of the macronutrient in the plant roots. Yes, those two questions.
Okay, fine. So one is you're asking about dilution effect. You're specifically asking about zinc. Okay, but I'll tell you in general what dilution effect is. So it is mainly because of this micronutrient only. जैसे स्टार्टिंग में हम अगर प्लांट की बात करें, हमारा प्लांट का बायोमास कम है और उसमें माइक्रोन्यूट्रिएंट का अमाउंट माइक्रोन्यूट्रिएंट ऑलरेडी कम है, ठीक है. तो माइक्रोन्यूट्रिएंट और प्लांट के बायोमास का अगर हम रेशियो देखें या फिर ऐसा देखें कि पर यूनिट बायोमास कितना माइक्रोन्यूट्रिएंट है, ठीक है, एक बात हुई. दूसरी बात, जब प्लांट ग्रो कर रहा है उस समय उसका बायोमास तो बहुत ज्यादा बढ़ जा रहा है, लेकिन माइक्रोन्यूट्रिएंट का कॉन्सेंट्रेशन कम ही है. तो इन दैट केस पर यूनिट ऑफ बायोमास माइक्रोन्यूट्रिएंट का अमाउंट कम है. दिस इज व्हाट वी कॉल डाइल्यूशन इफेक्ट. कि पर यूनिट बायोमास तो बढ़ गया लेकिन अमाउंट उतना ही है, तो वहां पे डाइल्यूट हो गया वो पर्टिकुलर न्यूट्रिएंट. दिस इज व्हाट इज डाइल्यूशन इफेक्ट. मेकिंग सेंस?
मैम ये सिर्फ माइक्रोन्यूट्रिएंट के लिए या सभी न्यूट्रिएंट के लिए?
मेनली माइक्रोन्यूट्रिएंट के लिए होता है, क्योंकि अगर हमारा बायोमास बढ़ रहा है तो मैक्रोन्यूट्रिएंट तो प्लांट को फिर भी अवेलेबल हो जाते हैं, तो वो उतना ज्यादा डाइल्यूट नहीं होता. लेकिन माइक्रोन्यूट्रिएंट के लिए मेनली है ये, क्योंकि माइक्रोन्यूट्रिएंट कम अमाउंट में है और बायोमास ज्यादा हो गया तो वो डाइल्यूट हो गया पर्टिकुलर न्यूट्रिएंट. ठीक है. दूसरा सवाल क्या था?
मैम ये जैसे आपने बताया डाइल्यूशन के बारे में.
मैम give it a brief idea about
plant uptake, no? You were asking. So there are three mechanisms of plant uptake we know. Root interception.
Hello, ma'am. Hello, ma'am. Hello.
Yes.
I hope there's Tamanna Sharma want to ask some clarification of the dilution effect. I hope the question is this question is asked by his in the section of the nitrogen. So I ask the question to you, ma'am. This question to you, ma'am. Actually, there is something problem in it.
Okay, if I'll send
Okay,
you
ma'am.
the clear
Please go ahead, ma'am. Please go ahead, ma'am.
Okay, Tamanna,
Okay,
another
ma'am.
question.
I send and then you send me, sir, ma'am, and I share to him.
Yes.
I send to.
Fine.
And ma'am, give the brief idea about the micronutrient transportation.
Uptake.
Ma'am, give the brief idea about the uptake of micronutrients.
So there are basically three mechanisms by which nutrients are taken up by the plant, either dilution, mass flow, or another one is root interception. So generally we cannot say which micronutrients, general term I cannot say that all the micronutrients are taken up in diffusion form or root interception or mass flow. They vary, means for different micronutrients, different mechanisms work. Wait, I'll tell you. Okay, this question I'll also tell, because in book also they have given macronutrients way of transporting. No information about micronutrient, but I'll tell you. I have noted both the questions. I'll tell. Any other?
Okay, ma'am. Okay, no problem.
Ma'am, please add me in WhatsApp group. Ma'am, I'll send in short.
I'll send to Suman sir and he'll.
Okay. Please, hello. Take me number 888099. Hello.
Sir, 1 minute.
880. I send in my number in the chat box. Please take this number and please give one personal message to me. I add you in the group. Don't worry. Okay.
Sir,
at that time you concentrated on the class.
Sir, in short, there is
okay,
no
I send. Please wait 1 to 2 minutes.
Okay, sir.
Okay. I send. You wait. Hello.
Okay, sir.
Silke, sir.
Okay, sir.
Hello.
Okay, sir. I received.
Hello, Dr. Silke, sir.
Yes, Suman.
Yes, sir. Class session is, sir, in the, so, sir, please give some feedback about this, sir.
Yes. Thank you, Suman. Thank you, Nidhi ma'am. You have given very nice information regarding all the micronutrients, along with all the explanations and all the deficiency symptoms and the methodology that is usually used for the acidic soil as well as the alkaline soil. You have given very nice and very informative information regarding all the micronutrients. I am thankful to you, ma'am, on behalf of all the course director, the faculty, and all the students. I am very thankful for giving us such a nice information. Definitely, this information will help all the students who are preparing for the NET, ARS, and GRS. Thank you very much, ma'am. Given very nice information, ma'am.
Thank you, sir. Thank you. It's really my pleasure. And yes, I'm thankful for this opportunity.
I have listened the whole your lecture. Very nice information. Very nice.
Thank you, sir.
And thank you for sparing your valuable time for this welfare.
Thank you, ma'am. Thank you, ma'am, giving this. Yes.
Thank you.
Suman, if there is no question.
Okay, sir. Yes.